very long-awaited lecture video. This has been a rough semester, so as you all know, I'm exhausted and very behind. But I did dress up for this lecture video. That's on predators today. This is one of my favorite dresses that I got in quarantine, and it is either my octopus dress, because it has 8 arms, because octopi are predators, or it's my tardigrade dress. Tardigrade are also sometimes predators. Some of them are herbivores. Or it could be my spider dress, which are also predators. And I also have on my special glow-in-the-dark tardigrade pin. If you're in the Monday group, you're about to see me in this outfit in person soon. And then I have some show and tell for today's video. One is some artwork I have hanging up in my house. This is a, I think it's a lino cut that is done by a friend I met living in Sonora when I guess in 2018 I was back in the Sierra Nevada for a roller derby tournament, and I met some artists up there, and one of them had done this kaiju drawing of a bark beetle, which we will talk about today, even though it's an herbivore. They are devastating the forests of the Sierra Nevada, so we'll talk about them today. This is the bark beetle as a kaiju. And then I also have a book recommendation. If you find predators cool and parasites cool, we're going to talk about the Red Queen hypothesis today. Is that backwards? Whatever. We're going to talk about the Red Queen hypothesis today. This is a really great book by Matt Ridley on the Red Queen hypothesis and how the evolution of sex protects, you can't even see my face, how the evolution of sex protects humans from the parasites that live inside of us. So, book recommendation, some art, themed outfit. I would also like to show you a predator that lives in my house. So hold on just a second while I go get them. This is my tiny little cat, Olympia. She's just had her 12-year adoptiversary. She's between like 14 and 16 years old. And she's a little predator that lives in my house. So you can see how, she don't like this. You can see how she has forward-facing eyes for hunting. She also has that reflective lens in her eye that I can't remember the name of that some predators have. And then she got those canines for shearing up meat. You're mad at me, aren't you? I love you. That's it. Now we're going to talk about predators, okay? Okay, bye. Okay, let's get to it. Let's talk about predators. Pew, pew, pew. Let's talk about the science of predation. And no, I guess I could mean the alien from Predator that Arnold Schwarzenegger battled, but mostly we talking about this kind. Predation can take a variety of forms in nature, and they function as agents of mortality in nature, which was a term from your textbook that I thought sounded like a death metal band, so I made that pretend death metal band a logo. Predation is the consumption of one living organism by another. That's the definition. Predators tend to be species that kill their prey more or less immediately upon capture. So let's define, this is a very general definition. Let's distinguish a predator from herbivores, parasites, and parasitoids. So predators usually kill their prey immediately on capture. If he gonna catch that groundhog, he gonna eat it right away. Typically, they consume multiple prey organisms, and then they continue to function as agents of mortality on prey populations throughout their lifetime. Herbivores are predators kind of in one sense, and we'll come back to this idea later on when we talk about bark beetles. But they typically only eat part of the plant. They don't eat the whole plant. They don't, well, in unusual circumstances, sometimes facilitated by climate change, they can kill the whole plant. With the exception of seed predators and planktivores, tiny, zooplankton tend to eat phytoplankton. In those rare cases, they're more like predators than they are like herbivores because they eat the entire organism and they eat it immediately. Parasites usually only feed on part of a plant or part of an animal, and they don't kill it because they're not a great parasite if they kill it. That they talk about that in that Red Queen hypothesis book that I showed you. And some are only parasites for a portion of their life cycle. So they only have a lot of parasites only have like a certain stage of their life cycle that's parasitic, and then they're free living the rest of the time. And that's what distinguishes them from predators. Parasitoids are an example of that. The free-flying adults are not parasites. And then the adult females will lay eggs on the host body, and then they slowly kill it over time. They don't kill it immediately. So that's a predator distinguished from all other types of feeding behaviors. Here's Laccaltera again. This is a classic model for understanding the interactions between prey and predator populations. It's a very simplistic model looking at the direct impacts of one prey population on another a single predator population. But it can be a useful tool, especially if you are working with a biocontrol agent that's a predator. I should have shown you that one that's in my house. Anyway, I am not going to ask you to use these equations on the exam. I just wanted you to show them to you. A lot of this stuff that they talk about with this model is like graduate level ecology stuff, so we're not going to get into it. But this top equation here is the model for the rate of change in the prey population. And I just want you to see the parameters that are involved here to just understand what's being modeled. So you've got included in this is the per capita rate of consumption of the prey, which is important. How much of the prey is the predator able to eat? The number of predators is also an essential part of this equation because they're linked to their prey population and will influence the prey population. The bottom equation here is the rate of change in the predator population. Of course, that's going to be informed by their ability to consume prey, the rate at which they consume the prey, and as well as the prey density, which is N prey. But this also includes so the difference between this and this is, well, there's lots of differences, but you can see here you've got a new parameter in here, which is the per capita birth rate of the prey and the predator are included. So, not going to have to use these. I just wanted to show them to you briefly. But what this model ends up giving you, and that's the most important part, is that you can see these coinciding fluctuations in predator and prey population. So, you see an increase in the prey population. The predator population responds in kind because its rate of consumption might increase with the change in the prey population size. But at some point, the predator population is going to reach a level at which it's consuming the prey more often than they can reproduce if there's no limit on the predator population. And so then you see a decline in the prey population because they're being overconsumed by the predator. That is then going to cause a later subsequent decline in the predator population because they have a lack of prey items available. And so both decline again. And then again, once the predator population starts to decline, the prey population can then increase its numbers again, and then you see these cycles, these shifting cycles with predator and prey populations. Now in the 1930s, Gauss, who we've mentioned before, wanted to do an experiment to see if this Laccaltera model actually holds up in an empirical way. So Gauss did lots of weird, cool ecology experiments with paramecium. So he did this experiment with P. caudatum, which we've seen before. In this instance, they were the prey item, And then Didinium nasudum is the predator. It's another small organism that eats paramecia. And he grew them in different environmental scenarios to see if he could get these population cycles that you see with the Laccaltera model between predator and prey. So this first example here is the P. caudatum and the D. nasudum grown in an oat medium without any sediment. So the P. caudatum have nowhere to hide from their predator, the Didinium. So you see this nice increase in the population. So he let the populations reach a nice size, and then he introduced the predator. And then when the predator was introduced, you can start to see a huge decline in the prey population. The Didinium eats so many, it completely causes that population to go extinct. And then because they have no food left, they also go extinct. So this experiment didn't work. So he said, what if I introduce sediment to this oat medium that I am growing them in, so that the Didinium won't go into the sediment, but the P. caudatum will, and they'll have a refuge where they can hide from the Didinium. Now, what happened with that experiment is that they were introduced at the same time. The P. caudatum mostly just hid in the sediment from the Didinium. They didn't live in the top part where the Didinium were, and the Didinium died out with the P. caudatum sizes grow a lot in the sediment. So that didn't work either. Now, the way he was finally able to get these predator-prey cycles that you see with the Laccaltera model is if he had multiple points of immigration into a population, so that the prey population was constantly being replenished if it was reduced by the predator population. So you can finally see these predator-prey Laccaltera type cycles when you have multiple immigration points of the prey into it, so that the predator doesn't just die out when prey population sizes are low. It doesn't just eat all of them until they're gone. You need to have this constant influx of prey items. And honestly, this is probably a more realistic scenario for like in the wild, because a predator can't realistically eat every single organism. Although with biocontrol efforts where you're like rearing a predator so that it can control a pest population, you kind of do want it to eat every single one of them, and then you kind of don't care after it eliminates the population. But in natural populations, a predator can't realistically eat every single prey item. So this is how he was able to get it. Karl Huffaker, who is an entomologist at UC Berkeley, did similar experiments trying to get these predator-prey cycles again in the lab with these predatory mites. So Typhlodomus is the predator. Iotetranicus is the prey mite. So this is a predatory mite that feeds on these. These are pests of orange crops. So likely Karl Huffaker, there's a lot of citrus pest management programs and money for that kind of research in California, and that's what when I was at UC Riverside, there was a whole group of people working on pests of citrus crops, insect pests. So great system, because it's easy. They're pests of the citrus, so he can grow them on the oranges and then introduce the predator to try to eat the prey item. So if he just introduced the prey onto a single orange, let that prey population grow, and then introduce the predator ate the prey until it died of starvation because it ate all the prey items, which is kind of what you want in a biocontrol scenario, but not what happens in the wild. So what Huffaker did is that he introduced complexity to the system, kind of like Gauss did with the sediment. So he got these population oscillations by introducing complexity. So he added barriers that stopped the predators from just following everywhere that the prey did. He partially covered some of the oranges so that the prey could get in, but the predator could not. And so there were refuges where the prey could hide. So Gauss got the fluctuations by adding immigration. Huffaker added complexity to the system and refuges where the prey could hide, and that's how he was able to get these population cycles. So this is likely how we see these cycles in nature. And then these models and this empirical data that supports these models shows a mutual population regulation between the predator and the prey. And you can have two types of responses. You can have a functional response, which is the relationship between the per capita rate of consumption and the number of prey. So this is a relationship between the rate of consumption increasing as the number of prey items increase. And then you can have a numerical response, which is with the increased consumption of prey results in an increase in predator reproduction. So numerical response has to do with the reproduction of the predator. A functional response has to do with the number of prey influencing how many individuals the predator can consume. And we'll talk about different ways that prey items defend themselves and have evolved to defend themselves from predators. So, there are three different models of functional responses to different increasing prey densities that ecologists use to look at how the dynamics of predator and prey populations. So what we're looking at here are the functional responses. So this is the relationship between the prey density to the rate of consumption. And so type 1, which these are two graphs showing the same type of functional response. So this is both type 1 functional response. So in this graph, you've got the number of prey consumed per predator, and this is just the proportion of prey consumed per predator. So this is just the total, and then this is the proportion. So with the type 1 functional response, each predator consumes a constant portion regardless of how many individuals. So they have some sort of like number that is the optimum number they eat regardless of how many individuals are in the population. So you see a steady increase in the number of prey consumed per predator, and then here you just see a flat level because this is the proportion. Then you have a type 2 functional response, predator functional response, and this is the predation rate decreases as predator satiation sets an upper limit. So this is actually a prey strategy that we'll talk about later on. There's also populations where predators can only eat so much of a prey item and then they get full and they can't eat anymore. I'm sure you've all felt like that before, especially during the holidays. So here is, you see a decrease in the proportion because this is the predator getting satiated by the population. And then here with just the total number, I think this is a little more intuitive, just the total number. You see a plateau where the predator starts to hit satiation and they can't eat anymore. They're too full. And then you have type 3, which is the predator rate decreases at low as well as high prey densities. I can't think of any great examples right now of this, but you see this dip here that indicates that the rate is low at low densities, and then you see a plateau here that shows that it's low at high densities as well. So that's why you see this fluctuation. So these are the three types of functional responses. And then these functional responses can relate prey consumed to their density. So one phenomenon that you can see that's related to prey density, and that is pretty common with a lot of species, is that predators will switch the prey that they feed on based on the density of that prey organism in the environment. So here is a model of prey switching here. So this is the number of prey in the environment, so increasing number of prey. This is the preference, so percent prey in the diet. You can see it's really low here, and then as the prey start to go up, and then you see a point. So they're feeding on it. So in this model, you've got prey species A and prey species B, and you see the predator switching over from one species to another species based on its density. So we're only looking at a single prey item here, but in this model, there's something else that the organism is eating because it's not eating as much of this prey species. So this is the line you would see if there's no rate of predation, there's no preference. So they just eat the same number regardless of how many individuals were in the environment. And then this is where you see prey switching. So you see an increase and a deviation from this straight line because once they hit a certain density, then the predator's like, oh, there's a lot more of these. I'm going to eat them instead. Here's an empirical study that showed prey switching. So this was looking at these sticklebacks and feeding them different proportions of Gammarus, which is a type of amphipod, and then Artemia, which are brine shrimp. So they were fed a mix of this Gammarus and the Artemia, and then they adjusted the number of Gammarus that were available in the population. So this would be low Gammarus relative to Artemia. This would be high Gammarus relative to Artemia. And you can see that this is the line where you would see no preference, and then you see that perfect curve where they switch over to feeding, having higher preference for Gammarus when the densities are high. So this is prey switching. Predators can also change where they are geographically in space in response to prey densities. This is an example with the redshank bird. Look at that little cutie with its little pokey feeding beak. And they feed on arthropod prey on shorelines. They tend to show an aggregative response, which is where they just kind of all aggregate. If you've ever been to the beach, you've probably seen shorebirds that insectivorous or arthropod feeding shorebirds that kind of group together in these big clumps. They just aggregate where there's lots of prey. So this is a look at one of their arthropod prey items and the density in the environment where these redshank birds live. And at low densities, you see not a lot of redshank birds, but at high densities, you see this exponential increase in the number of redshank birds. So they all kind of aggregate around where there's lots of this arthropod prey. So that's the aggregative response. Now let's talk about optimal foraging theory, which is a really cool idea. And we talked a little bit about when we read the killer whale paper, which I found super fascinating. I didn't know much about orcas at all until we read that paper, and they're just like, their cooperative feeding behaviors are so cool. But you might see them again in relation to optimal foraging theory on the exam. Hint, hint. Okay, so let's talk about optimal foraging theory. This is the idea that natural selection is going to favor the most efficient foragers in the environment, and so behaviors or preferences that allow them to receive the most nutrition related to the energy they spend finding that food, those organisms are going to be favored. They're going to reproduce more in the next generation, and so they heritable traits that encode for that preference and that feeding behavior are then going to be passed on more readily to the next generation, and that's going to cause the evolution of optimal foraging strategies for a given prey item. So what you see with this is that there's a balance between the time and energy that you spend trying to find or handle food versus the nutrition that's gained from that individual food item that you're eating. We talked a little bit about that in the Monday group and then online with the Friday group about the orca whales only feeding on those Wendell seals. So there's a lot of cost involved, but theoretically, and I don't even know if anybody knows this, maybe those Wendell seals are extremely nutritious, so that cost really is worth the benefit. And so you can see these costs and benefits that are weighed by organisms and their decisions on what to feed on in the environment can influence, they're going to be based on diet composition, and they're going to influence diet composition and the choice of organisms in the environment. And when we think about the time spent foraging, we want to think about the time they spend not only searching for the organism, but how long it takes them to actually like process it and eat it. Maybe, oh, it's too cold now. I got some good predator videos for you to watch on Schoology. But this is a really cool experiment looking at prey choice in the pied wagtail bird. That's a dude there. Look, he's eating right now. So they have their potential prey types in their environment are sarcophagid fly, dung-eating animals. So you got scarab beetles and these sarcophagid dung flies, which I think are kind of pretty even though they eat poop. They have these potential prey types in the environment. This is the available distribution of the prey size in the environment. In wagtails, which you can see here, this is the available prey, and this is their preference. So it doesn't exactly match the distribution of the prey. They prefer something a little smaller than the mean size. So they show a decided preference for medium-sized prey, and the size of the prey corresponded, what the researchers found, to the prey the birds could handle most profitably. So basically what they did, looking at the calories per second, you'll have to read the tag. I can't remember how they calculated this. I'm sure they did like nutritional analyses on individual insects based on their size. Oh yeah, that's actually probably exactly how they did it. They just ground up the bugs of different sizes and then looked at the calories, and then they watched the birds and saw how long it took them to eat bugs of that prey size. So there's very little nutrition in these small organisms, and so the birds basically ignored them because even though they were easy to handle, they weren't worth the payoff. It's like eating a rice cake. They just didn't return sufficient energy for them to really bother. But on the other hand, these large prey, so it looks like you kind of get, and then so this is calories per the time it takes to handle them. You see a dip because these large prey it just took them way too long so they like throw out a big one, and then it just took the bird way too long and too much effort to try to figure out how to fit it in their mouth. And so the optimal calories per how long it takes them to eat it is about the same size as the ones they prefer in the environment. I love this kind of stuff. I think it's really cool. But sometimes the hunters become the hunted. Da, 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 da. This is just a short section of your book chapter, but they talk about two species of tit birds, the crested tit and the willow tit. They're both insectivorous birds. Mostly, I think they just feed on insects and maybe some seeds and things. So they're considered predators, insectivorous predators, and they feed inside these coniferous trees. And they also live in the same environments with these predaceous Eurasian pygmy owls. Now, in normal, in like normal circumstances, quote unquote normal circumstances, these pygmy owls will usually feed on voles if the vole population is large enough. But if the vole population decreases to a point where they have trouble finding them, they will switch over to feeding on these two tit species. And what the tit species will do is they have a behavioral modification based on this. So if they start getting hunted by Eurasian pygmy owls, they see other birds starting to be hunted, everybody starts disappearing from the population. What they'll do is they'll start foraging on the insides near the trunk of the tree so that they have better hiding in the foliage rather than feeding in the open areas and around the edges of the branches. So this is one predator influencing the feeding behavior of a different predator. And then I'm sure you've all seen it. Well, I don't know if you spend as much time on the internet as I do, but this is I'll put a link on it because there's this really cool study about how climate change has made these great tits in Europe become like terrifying, vicious, murderous predators. And I just love this photo. He looks like he's like posing with his deer. I think I saw somebody on the internet said that this would be this bird's Tinder profile of him like with his mouse that he caught and killed. I'll post a link. It's a really cool study about predation by great tits in response to climate change. And so with these, the pressure of the predators, that the predators are constantly influencing their prey populations, and then the prey populations are constantly having to adapt to that. This is where you get the Red Queen hypothesis. So if you've read Through the Looking Glass and Alice in Wonderland, the Red Queen in that book is, she's constantly running, and the quote is from the book is, "Now here you see, it takes all the running you can do to keep in the same place." And so that's why this is called the Red Queen hypothesis. It's named after the Red Queen from Alice in Wonderland. So prey items are constantly in an evolutionary arms race with their predators to try to evolve better defenses against predation, and then the predators are responding. So you're constantly having to run in place, and the forces of natural selection are constantly causing new adaptations to predators. So you're constantly running in place to make sure that the species survives. And I thought this garter snake and newt tetrodotoxin study from a previous chapter was a really great example of this. So in some of these populations, you can see like these newts are hella toxic. Like look at how, like that's greater than 90. That's like a really toxic newt. But the garter snakes that live there have evolved really high resistance to this toxicity in that environment because of this heavy selection pressure that must be their only prey item. And so the garter snakes have evolved really strong defenses to the toxin, and the newts are evolving even stronger toxins in kind. So great example of the Red Queen hypothesis. Now let's talk about one of my favorite things, which is how prey items defend themselves against predators. They can use sometimes chemical defenses. I've included a video online. This is a bombardier beetle. This is a beetle species that shoots hot, boiling acid out of its butt at its predators. Like that smoke is like legit smoke. It's boiling acid that they shoot at their predators so that they don't get eaten. This is something that has evolved through very, very strong selective pressures in the environment, so that they need very strong predator defenses for this species to continue. So watch that video on Schoology. Let's go through some of my favorite predator defenses. One is basic, just cryptic coloration. I think this is a flounder fish that's blending in with its environment. So just matching the background of your environment, or even the thing that you're eating, so that a predator can't detect you visually. There's also object resemblance. This is a leaf bug resembling a leaf, so predator doesn't think to eat it. I guess y'all haven't seen my Halloween costume, but if you go to the SVC biology page, you'll see one of the costumes I wore last week for Halloween was an owl spot moth mimicry dress that has these big sleeves that look like a moth that's mimicking the eye spots of an eye. So that would just be basic mimicry or object resemblance there. There's flashing coloration, so flashing a little white butt to confuse a predator. There's warning coloration or aposematism. This is my favorite, obviously, because bees have it. So this is a striking, visual, easy to remember pattern that lets a predator know, I don't want to eat that. So the way that this works is that a predator has to try to eat something before it learns this signal. So predator comes up, tries to take a snack on this bumblebee, and gets stung in the mouth and is like, ow, ow, that hurt. I don't want to eat anything with black or yellow stripes again. So there's learning that's involved here with aposematism. And then with aposematism, you can get Batesian mimicry or Mullerian mimicry. Hopefully you remember this from Gen Bio. Batesian mimicry is when one non-toxic or hurtful organism mimics a hurtful one. So this is a honeybee. It has the brown and yellow stripes. If an organism eats this, it will get stung. Hopefully it will learn that pattern. later in the season, hoverflies come out, and they look a lot like honeybees. And so what they're banking on is that the predators learn the signal from the honeybees earlier on, and they're like, oh, I'm a honeybee too. Please don't eat me. But they're not toxic, so if they eat them, it'd be a good snack. So this is Batesian mimicry, non-toxic, mimicking a toxic. Mullerian mimicry, which is best exemplified with heliconius butterflies in the Amazon, is when multiple toxic organisms converge on a single signal to tell a predator not to eat them. So this reduces the amount of learning a predator has to do and reduces predation on multiple species. So each of these pairs that you see here are two, I don't know which is which, but it's probably Heliconius aerato and Heliconius melpomene. These are two different species. That is a different species than that. That is a different species than that. That is a different species than that. That is different than that. And they form these mimicry complexes in the tropics. I could talk about this all day. I love mimicry, you guys. They form these mimicry complexes in the tropics so that a predator only has to learn a single warning signal. And likely they form these different patterns in different regions of the tropics because there's different bird predators there that are learning different visual cues. Anyway, if you want to talk about mimicry, ask me later. They can also sometimes form protective armor. This is one of my favorite, most disgusting examples. This is a beetle larva that has covered itself in its own feces so that a predator is like, ew, I'm not going to eat that. It's covered in poop. Another great example of this is the spittlebug that we talked about. So we talked about them in herbivory because they eat a very low in nutrition food, which means they have to eat a lot of it. And then that spittle, that foam that they eject out, helps protect them against predators. There's also behavioral defenses, like a possum playing dead. You don't want to eat an animal that's already dead if you're a predator. And then predator satiation, which which I mentioned very briefly in like 3 slides ago. So this is when a prey population has such high prey density that they cannot possibly all get eaten by the predators. Cicadas are a perfect example of that. Periodical cicadas, they come out in such vast numbers that a predator can eat all they want and they still wouldn't be able to eat all of the cicadas in the population. Mayflies are another great example of this. They come out in huge numbers, so large that not all the predators in the world could eat all of them. Some of them can have permanent defenses that they are born with, so like venom and a bee sting. Okay, that's all I can think of right now. Well, actually, a lot of these are just born with it. Maybe she's born with it. Maybe it's permanent defenses. And then induced defenses is when they get attacked by something and then they do like a warning display. So actually, Joe Jr., if you saw that video I posted, that's actually a great induced defense. Joe Jr., before Joe Jr. pupated, I made this video. Well, so it's permanent defense is looking like a snake, right? So predators don't eat it. Its induced defense is when I poked it, it stuck out its little osmeterium that looks like a snake's tongue. It was like, I'm a snake, don't eat me. So, that was a great example. Go me. Okay. Predators can also, so like we talked with the Red Queen hypothesis, predators can evolve really cool hunting tactics to get around or to trick their prey. And I've included a really awesome video about these Photurus fireflies that eat Photinus fireflies. It's one of my favorite examples of a predator hunting tactic. They're called femme fatale fireflies, and if you watch the video, you will learn why. It's cool. And then in some ways, depending on the type of herbivore, they can prey on autotrophs and function like predators. Call back to the beginning is that bark beetles especially in places now, with the interaction of climate change, they have started to function like predators on conifer populations in the Sierra Nevada. So with climate change, California has been seeing many, many years of drought. That drought has weakened the immune defenses of the trees that live in the Sierra Nevada. And with weakened immune systems in these trees and defenses, I shouldn't call it immune defense, it's a tree. With weakened herbivore defenses, bark beetles have come into these trees and have devastated and just killed them all. In normal circumstances, in non-drought circumstances, they do not do this to these trees they live inside of. So they make these tunnels throughout the trees, and then sometimes they bring a fungus with them too. In normal circumstances where water is not limited, these trees can defend themselves, but when they are already stressed due to drought, the bark beetles just kill them, and you can just see whole swaths of the pine forests in the Sierra Nevada that have been devastated by bark beetles. So we'll talk about, okay. Sometimes though, plants have evolved protection from their herbivore predators, quote unquote predators. This is a bug this is actually, thorns protect plants, but this picture is kind of a lie, because what probably happened is that this bug was put onto this by a shrike. Shrike are these birds that use plant thorns to skewer insects and kill them, and then they eat them. So that's probably what happened here, but thorns can be a defense against herbivores. This is a thorn here that's a defense. This is, which you learn about in Gen Bio, this is the ant acacia relationship, mutualistic relationship. So the acacia plant provides a home inside the thorns, and then it provides these extra floral nectaries as a food resource for the ants, and then the ants defend the plant from scale insects or other herbivores that try to eat the acacia. Trichomes, so anytime you find a hairy plant, those trichomes are meant to deter herbivores. I noticed that I've never had a garden until this year, but I noticed in my garden that my cucumbers and my melons all were super, super hairy when the fruits were developing, and then as they got bigger, they lost the trichomes, which I thought was a really cool herbivore defense mechanism to protect them before they're fully grown and their seeds have developed. Super cool. And then we'll talk a little bit more, but I just wanted to introduce the concept now that through food webs, carnivores can also affect plant populations. We're going to talk about this example of wolves being reintroduced in Yellowstone later on in the semester. But that's it. That's it for predation. Stay tuned for the next lecture. Bye-bye.